morning, everybody. My name is Marzieh. I'm a postdoc fellow at University of Alberta, and my supervisor is Dr. Graham Palasto. And this work that I'm presenting today is also co-authored by Dr. Val Kearney and Professor Martin Groenen from Wageningen University in the Netherlands. As you can see, the title of my presentation is Genomic Diversity and Relatedness in Heritage Chicken Lines using Whole Genome Sequence Data. So let's give a brief introduction first. In the past 50 years, there has been a steep increase in the demand for poultry products. And the increasing of the production has been met along with the genetic selection for improved growth, efficiency, health, and reproduction. Generally, selection tends to reduce the number and type of genetic resources contributing to the majority of the production. However, at University of Alberta, we have 10 heritage chicken lines which haven't been subjected to the same intensive selection pressure as the commercial lines. And these heritage lines may have genetic variation that has been lost in commercial breeds. And in a study by Muir et al. In 2008, it has been shown that around 50% or more of the genetic diversity in ancestral breeds is absent in commercial pure lines. So these heritage lines can be valuable source of genetic variation. However, in these chicken lines, there is a high inbreeding, and so there is a risk of loss of genetic variation due to inbreeding because the selection in these lines has been just random. It means individuals have been selected randomly to mate to each other to produce the next generation, and that's why the inbreeding among these lines is high. For the conservation and management of these heritage lines, the first step is to assess their genetic diversity, which leads to our objective to explore the patterns of genetic diversity and relatedness among heritage chicken lines using a standard population genetic analysis. In this table, you can see the chicken line name and some information about the chickens. The first line is the name of the lines. In the brackets, you can see the short names that has been used in this study. We have lines 20s, 30s, and 90s, and BL as brown leghorn, and LS as light Sussex, NH as New Hampshire, Sask BR as Saskatchewan Birdrock, and Shaver BR as the Shaver Birdrock, and the Rear, which is the Shaver Rhode Island Red, and WL, which is the white leghorn. And four of these lines are mid-type, as you can see, 20s, 30s, and 90s. And New Hampshire lines are mid-type, and the rest of the lines are dual-purpose type. And in the third column, you can see the average sequence coverage of these lines that have been sequenced. On average, the coverage, the sequence coverage is 10, which is good and shows the quality of the sequence is fine. And the sample size is total for the total number of individuals which have been sequenced are 71. And in the last column, you can see the original geographic location of the lines. So in total, as indicated, 71 male chickens were sequenced to be used for this study. So when we have the sequence the individuals, we need to do some steps in order to call the variants that are with good quality, and also we should do some quality controls on the back of called variants. We used several software. As you can see, I wrote the name of the softwares in red. Thermomatic, BWA, Picard, and GATK in order to trim the sequence rates and do alignments and the variant calling and the quality control. So we ended up having 16 more than 16 million SNPs, which are the single nucleotide polymorphisms, which are the genetic variants, and we did some quality control. As you can see here, I listed the quality control criteria for the sequence variants. And then after doing such quality control, we ended up having more than 13 million SNPs or genetic variants remained for the 71 male chickens across the whole genome. A final step of quality control were also done on called variants, and these are the criteria for the quality control and exclusion of the SNPs. And finally, we had more than 5 million SNPs on chromosome 1 to 28, which are the autosome chromosomes for chickens for downstream analysis. Several approaches have been used to investigate the population structure among heritage lines and also assessing the relationships among these lines. And the first one is genetic differentiation, the second is admixture analysis, and the third is principal component analysis, and the fourth is phylogeny analysis by making a neighbor joining tree. And also estimation of the inbreeding coefficient has been used to assess the relationship among the 10 heritage lines. I will show the result of each analysis in the coming slides. For assessing the genetic differentiation among the 10 heritage chicken lines, we use the measurement called fixation index, or FST. Here in this table, you see the FST values which is based on the pairwise population comparison. And in the very top row of the table, which is separated from the rest of the table, you can see the average FST values, which is the average of FST values of one line compared with the other lines. And based on these values, you can see that the FST values range from 0.1 to 0.45. And the green in the green part of the table, it shows that the bill-like horn seems to be the most genetically diverse line compared with the other lines, and line 20, which is shown in yellow here, has the lowest average FST, which shows that this line 20, is the least divergent line compared with other lines. Principal component analysis is another approach to assess the relationship between the heritage lines. As you can see here in this picture, there is none of the lines dispersed across the plot, indicating that there was not large variability within any of the 10 heritage lines. Close to the middle of the plot, as shown here, there is lines 20s and 30s, which makes a cluster together as expected because of their similar origin. Here in this part of the plot, you can see that lines mm, Sugar BR and Sask BR grouped together at this left side of the plot, and here at the right side of the plot lines New Hampshire, Rear and Light Sussex also clustered together at this position. Lines BL and Brown Leghorn and WL White Leghorn are also on the left side of the plot on the top. You can see them. They are close together, but they are not making a cluster. Another approach to assess the genetic structure of the heritage lines and their relationships is to analyze the phylogeny analysis by building a neighbor joining tree. As you can see here, the results of the tree neighbor joining tree shows that for these three clusters, and that these results are consistent with the previous results shown in the principal component analysis figure in terms of the genetic distance and relatedness of the lines. However, there is a surprising results here in the blue circle. You can see that the line 90s seems to be closer to the dual purpose lines brown and white leghorns than to the mid-type lines, which are lines the 20s and 30s. Our expectation was to see the closer relationship between lines 20s and 30s with the lines 90. However, based on the establishment of the line 90 in year 1957 from dual-purpose breed, which is white polymoth rock, we hypothesized that in year 1957 line 90 was genetically closer to their dual-purpose founders, such as Leghorn breeds. But later on, from year 1957 to 1978, the selection objective was more towards meat production and therefore production of meat-type birds. The inclusion of the meat-type genetics from crossing between line 19 and other meat-type lines may justify the distance between line 90 and the two other meat-type lines, which are line 20s and 30s, and instead being closer to the Leghorn breeds. To assess the genomic diversity among the heritage lines, we use the following genetic diversity measurements, including the expected heterozygosity, observed heterozygosity, proportion of polymorphic markers, and nucleotide diversity. I'll show the results of genomic diversity in the next slide. So here in this table you can see the genetic diversity measurements results. As you can see here in the green arrows, you can see that for lines 20s, 30s, and 90s, the inbreeding is lowest, so the proportion of polymorphic SNPs, observed heterozygosity, expected heterozygosity, and nucleotide diversity are highest, but in the red rows, which belong to lines brown leghorn, light Sussex, New Hampshire, and white leghorn, the inbreeding rate is highest. And so when the inbreeding rate is highest, that means that the nucleotide diversity, observed and expected heterozygosity, and proportion of polymorphic SNPs are lowest for these four lines. From this study, we have some conclusions. So we characterize the genetic diversity of the heritage lines maintained at the University of Alberta using whole genome sequence data. These genetic diversity results helps our understanding of the genetic potential of the 10 heritage chicken lines. And according to our results, lines 20s, 30s, and 90s have sustained high level of genetic variability. However, some of the other lines, including the brown and white leghorns, light Sussex, and New Hampshire, showed high level of inbreeding, which needs a special attention. And to maintain the genetic diversity, we need to set up an optimal breeding program for these lines, and such a breeding program can be performed by selecting the lines in order to minimize the average kinship between selected parents. It means controlling of inbreeding rate and maximize optimal genetic contributions of parents to future generations. Finally, I would like to thank all of you for your attention. And if you have any questions about my presentation, please feel free to send me emails. And here is my email address. I will be happy to answer you. Thank you.